thanks for coming out today to our Darwin Talk. Today we have Dr. Chantel Sloan with us. She grew up in Connecticut. Oh, is that true? Okay. She grew up in Connecticut, and she got her bachelor's in biology at BYU, Hawaii. And then she went on to get her degree, her PhD in genetics from Dartmouth. She went on to do a postdoc at sunny Stony Brook, and then another postdoc at Vanderbilt University. And she's currently an associate professor in the Department of Public Health at BYU. She teaches health, disease determinants, and medical geography. She works on epidemiology of respiratory viruses, including the spread over space and through time. She has published papers in Journal of Infectious Diseases, Health and Place, Influenza, and has been an active contributor in the local COVID response. She's written articles for the local news outlets, including an op-ed for Deseret News on the balance of considering personal liberties and what's best for the common good with regard to mask wearing. In short, you should wear a mask. And she's an advocate for COVID policies at BYU and other institutions of higher ed. So please join me in welcoming Dr. Sloan.
Okay, working okay? You've been fiddling with it a lot? Great. Good to be here. Thank you so much for having me. This is my first time really on Snow College campus, and I am impressed and loving it, and it's beautiful. It's great to be here. I mean, we have kind of a mixed audience today, people with different scientific experience levels, and so also because we're a smaller group, even though it's being recorded, if there's anything that I say where you're going, oh wait, I want a little more detail there, or can you explain that again, or I didn't catch that, feel free to interrupt me. I'm very used to it. When I teach my classes, I tell my students that the only real rule I have is that you're not allowed to start a question with the word sorry, because we're never sorry about having questions. Okay? So don't be sorry about having questions in here either. Okay? It can be a conversation. All right. Now, what I didn't check is if this will. Again, so I do teach in the Department of Public Health. I have a genetics background. I've worn many hats over time. I've done a lot of research on influenza, now on COVID, also pneumococcal pneumonia, and respiratory syncytial virus. I also do some air pollution work, but I think we're all here for respiratory infectious diseases today, so that's where we're going to focus. And I was invited to talk, I love to cross my genetics and public health experience, and that's what I was invited to talk about, so I was really excited. And I want to talk to you about the genetics of SARS-CoV-2, right, the virus that causes COVID-19, from the perspective of how do these genetic changes impact health outcomes in real time. Okay, so we'll back up a little bit, talk about where SARS-CoV-2 came from, what we know about it, and where we're going, and how these new variants that we're all hearing about in the news right now are going to impact the public health response going forward. Okay? Okay, so where does SARS-CoV-2 come from? There are a variety of possibilities. SARS-CoV-2, COVID-19, is what we call a zoonotic disease, which means it originated in another animal species besides humans. The vast majority of diseases experienced by humans are zoonotic, so this is not a new phenomenon. Okay? When we think about influenza, MERS, SARS Part 1, Hendra virus, I mean, we could just list off most viruses that influence human populations, and we can trace them back to an animal of some kind. There's a few that we can't. Respiratory syncytial virus is one of the few, along with some rhinoviruses, where humans are the original host, but the vast majority come from somewhere else. The hypotheses about where SARS-CoV-2 came from are a little bit varied, but we're pretty sure at this point that the original source, what we call the reservoir, is this horseshoe bat, this cute little guy right here. A lot of our diseases spill over from bats and birds. Spillover is, let me back up, see, I used vocabulary words, I'm finding it. Spillover is that moment in time where a disease moves from one species to another. So, and then that disease may or may not emerge as a new epidemic or pandemic in humans. Okay, but that moment or the series of moments where that's where the only one's infected, now suddenly we have some humans infected is spillover. Okay. So there's one hypothesis where, there are horseshoe bats interacting with humans, probably because people were hunting and eating wild game in China, just like they do around the world, including the United States. Okay, and there was contact and the virus spilled over. The most probable one, the strongest hypothesis right now, is that it went from bat to some kind of intermediate species. Remember, there's always talk about the pangolin at a food market to humans, but it could have also been cats. There are a variety of things it could have been. So that spillover could have happened multiple ways. And then there's another option where there's another human step where it went from bats to potentially an intermediate species and then got introduced to the frozen food chain, supply chain. And that's how it got introduced to Wuhan. And it may have been around for a while before Wuhan, but that's such a major populous center that when it hit there, it really took off, and that's when it got on people's radar. Right? So a variety, but we're pretty sure initial spillover involved our furry little flying frontier. Okay. So let's now dive in and look at the Virion particle itself. What's really going on with this little virus? Viruses are very simple biological packages. Okay, they contain some kind of genetic material, either DNA or RNA. SARS-CoV-2 is an RNA virus. We'll talk about the difference in a second. And it has these proteins that surround it. There's a protein that surrounds the genetic material. And then there's also structural proteins and a membrane that form around it, hold it inside like a little ball. And then we have the often talked about and infamous now spike proteins, which sit on the outside of the virus and are really, really essential for how it binds to our human cells, and the virus is able to infect us and cause disease. Okay. I also want to mention that it can code for and contain these little proteins up here called polymerases. Polymerases are for copying. So when the virus infects our cells, it wants to make many, many copies of itself. So it'll use polymerase, that's a little machine, to make multiple copies of its genetic material. The inside, where the RNA is, it's very organized. The RNA loops and turns on itself in a really organized way, much more organized actually than some other viruses, which is kind of interesting. But all in all, it really is just a simple little biological machine that just wants to get up there and reproduce. So, and it operates by some pretty simple rules. Okay, so just to back up a little bit, talk a little bit about the genetic material we're working with, which in the case of SARS-CoV-2 is RNA. Genetic material codes for the different structures, the proteins, the different actions that are happening in our cells every millisecond of every day, right? So DNA is what's in the nucleus of human cells. I like to think of it as imagine a library, and there's a really old copy of a book, like an original cookbook from medieval times, and you want to make a recipe of it because you're practicing, maybe you're going to have a Great British baking show or something, and you want to practice this, and so, but you can't take the book out of the library, it's too precious, too fragile. So you want to make a copy of it, a photocopy of the page, And that's like going from DNA to RNA. It's like making a photocopy. And you can do whatever you want with that photocopy. I mean, you can spill food on it, you can tear it up, you can make more copies of it, right? It's not as essential. Okay? But it's still helpful. Without that copy, you wouldn't be able to make the item, make the recipe. Okay? So in our cells, we have DNA in the nucleus, but we also have lots of RNA running around and doing different jobs. Okay? This kind of single-stranded copy. And that's the type of genetic information that we find within the SARS-CoV-2 virus. Okay? Any questions so far? No? Okay. You're all like, we know this. Go on. Okay. Okay. So where do new variants come from? This has been in the news quite a lot lately, that we have these variants of COVID that might change our health response, how we think, what we do with vaccines. Okay? But where does that come from in the first place? When the virus, SARS-CoV-2 in this case, enters our cells, it makes many, many copies of itself. So that polymerase I was talking about before goes in and copies the RNA, and some of the RNA goes to our cellular machinery to make proteins, to make those spike proteins and things that are going to go on the outside of new viruses, but others just make other copies of the RNA to go inside the new viruses. Okay, so we have a couple different things happening. And that polymerase, that little biological machine that's a photocopier, is really quite good at what it does, but it's not perfect. Every now and then, on the order of between 1 and 2 times 10 to the minus third per year at each site, okay? A really small amount of times per each spot on the RNA, okay? It makes an error. And it replaces one bit of genetic material with another, okay? Because it's not a perfect machine. And in some cases that error can get corrected, but it doesn't always happen. The vast majority of times that there's a mutation like this, it doesn't matter. It doesn't matter for the virus, it doesn't matter for us, it just kind of comes out in the wash. Right? But every now and then, it does matter. Either it really matters for the virus because now it can't replicate as well, or it really matters for us because it can replicate better, or transmit better, or overcome us having been exposed to the virus before. Okay, so all of these variants in the RNA can result in variants on the virus itself. So maybe in that spike protein, things look a little bit different. Okay? Okay. So this mutation rate that I told you is kind of 1 to 2 times 10 to the minus third per site per year, right? This very small number. Isn't the same everywhere in the virus. There are some places that mutate more slowly and some places that mutate more rapidly. And that's because some places within that RNA have more evolutionary pressure on it. Okay. If a mutation is beneficial and it occurs in one of these key regions, then the virus can do much better at replicating and reproducing. Okay. So they found that the spike protein mutates a little bit faster, the polymerase can mutate a little bit faster, and some of those structural proteins can as well. We also found that over time, if you look at when we first had that big outbreak in Wuhan, through those first 4 or 5 months, there was a really high rate of mutation, and then it kind of slowed down a little bit. It's still happening, but the rate changes over time as the virus kind of figures out humans. It'll coalesce and say, you know what? We've tried changing this part of the spike protein lots of times. Amanda's more excited a little bit, but this part of the spike protein has changed many times. It never confers anything beneficial. Right? The mutation kind of slows there in that region, or we see the RNA code start to stay the same in that region. So this isn't a hard and fast target where we always know exactly how much everything's mutating and at what site. It's changing over time, it's changing by location, and it depends on what part of the RNA we're working with. So a whole lot of variability, a ton of unpredictability, right? Because it's all random unless it makes the virus better or worse at what it's doing. Okay. So here, not study, but this is a phylogenetic tree of many thousands of genetic copies of SARS-CoV-2. You can see each of the tiny lines coming off is one sample, and then they've grouped them into these genetic clades or subgroups. They say, okay, these samples probably all have the same ancestor. Okay, so even within this short amount of time, just a little over a year that we've known about this, it's diversified in all of these different ways. Okay, it's diversifying at about the same rate as some similar RNA viruses. Okay, so it's not doing this at a rate that's really surprising, but it is fast enough for us to worry about. Okay, so of all those different variants that I just showed you on that phylogenetic tree, most of them are kind of a moot point so far. They're variants that don't really change the way that SARS-CoV is transmitted between people or how serious our infection is. But we do have a few variants that have popped up that are a concern. These are the ones CDC has currently listed. So we have the UK variant, and I'll just call them by their countries, but I'll tell you their technical names too in a second. But the UK variant, which we've heard a lot about in the news, right? The South Africa variant, which is a pretty new one, and the Brazil variant, which is probably the most concerning one, personally. We're still gaining information about all three. There's also another variant that's just come out of the Bay Area in California. We don't know as much about it, but it's not quite listed by CDC as a variant of concern yet, so I didn't include it. But just so you know, it's happening in the US too. It's just a random chance of where and when this is going to happen. Okay, so let's talk about the UK variant. The UK variant includes many mutations. The technical term you've probably heard for it the most is this B.1.1.7, okay? B117, just under the title there. It has a lot of mutations, many of which are in that spike protein, that really essential exterior protein where SARS-CoV-2 binds to our human cells and enters, okay? We've seen it spread worldwide. This variant is now found essentially everywhere. It was contained for the UK for a while, but pretty inevitably ended up here. It might increase risk of death compared to other variants. The jury's still out on that a little bit. It depends on which study you read, because there's some evidence that you might see an increased rate of mortality with this one. It definitely is more contagious, so it confers higher transmissibility between people. So that's why when it introduced, we kind of saw some spikes in transmission, increased waves of COVID-19. Okay. No known effect on vaccine efficacy, which is great news. So far, Moderna and Pfizer have looked at efficacy. If you're infected with the UK variant, it doesn't change. So that's good news. It has been in the U.S. since the end of December of last year. 2020 is the year that shall not be named. All right. So the South Africa variant, this is a little newer. Okay. We know a little bit less about it, but we do know some interesting things. It shares some of the same mutations with the B117 from the United Kingdom. Okay, so there's some overlap. It emerged separately, but some of those same mutations show up. Many cases outside of South Africa. It is not contained to South Africa. There's some evidence that it increases transmissibility. Like the UK one, it seems like so far, The vaccine is doing just fine fighting it, so we don't think there's any major changes there. But there is a lot that we still have to learn about this variant. Still a lot going on in researching it and doing disease surveillance. So with the Brazil one, I said the Brazil one's probably the most concerning. So this one's the most common reference number is P1. Okay. Variant P1 has 17 unique mutations, including on that really essential recessive protein, the spike proteins. The biggest problem with this one, and you probably heard about this in the news, is that it seems to be able to reinfect people who were already previously infected with COVID. And this is very pronounced in the area of Manaus, Brazil, where it's thought that there were about 75% of the people who lived there were infected with COVID already. They already had COVID-19. And so they were at such high infection rates, and they saw so much death. It's been really tragic what's been going on in that district. But they thought they were reaching herd immunity, this level where you're protected from future outbreaks. But then this variant showed up, and suddenly we had all these people being reinfected, which isn't great news because as it spreads globally, right, which is pretty inevitable, we can see surges of cases in places where things have been calming down. Now it's possible, talk about this in a second, that the vaccine will still be okay for some reason, still confer some protection, and Manaus isn't an area that's seen a lot of vaccination. So there's still a lot to learn about this one as well. So I don't need to say, oh no, the vaccine's toast, because the vaccine is not toast. But it might require some boosters, it might require just being a little careful if this particular strain is around, or variant is around. We're learning a lot about it. And some people have suggested the Manaus city has some problems, but anyway, the principle I think holds up, that they're seeing a lot of reinfection. Okay. So this figure is from this really great paper, it's Cordary et al. From last year. I really recommend it if you're interested in understanding how viral variants travel around the world. It's just chock full of great images. And I just snapped one piece of one, actually. But what they did was they took samples of SARS-CoV-2 from around the world, and they compared just this one mutation site. Okay, so the original variant that came out of China, you can see the kind of the lower right-hand corner there, it's orange, and that's called D614. So the original is D614, and that just means that there's a change in the protein at spot number 614. Okay. Then this new variant that came up earlier last year, okay, is G614. We went from D to G, right? And you can see that prior to March 1st, you take a look at panel A there, whole lot of orange, right? That Chinese Wuhan strain or variant, same strain, variant, is predominating in most places around the world, except in Europe. You start to see this second variant, this mutation pop up, where we go from D614 to G614. But then look after, or toward the end of March, right? This is not a long time window. Okay, just a few week time window, we go from D being predominant to G being predominant in every location worldwide. Okay. Now this was also when, except India. India still had more original. But you can see things in China were slowing down quite a bit. They don't have as many samples from there during this time window, so we're not sure how much of G614 was there, but we can see that pretty much everywhere else there's this very rapid transition from one variant to another. So that's why when I say, oh, we've got the South Africa variant, the UK, the Brazil, and it's probably going to spread really quickly, it's because that's just how it works, right? If you have a variant that makes something more contagious, that can become the predominating variant in a matter of weeks. We have to keep really close track of it and do a lot of disease surveillance. Okay. So let's talk a little bit more about how and why this evolution is going to impact the efficacy of our vaccines. Okay. I know some of you have been vaccinated already. Congratulations. I wish I were among you. I'm not. I probably won't be for another month or so, but that's okay. I'm not top priority for good reason. But for me, watching all of this and being involved with the COVID response on some level, the vaccine coming out has felt like this light at the end of the tunnel, right? And I still think it is. But these variants are kind of throwing a little bit of a complication into what we do and how we move forward. Okay. Real quick, just to get everybody on the same page, I want to talk about our immune response to viruses, what happens when a virus infects us, and what our body does. Okay? If you've gotten the vaccine, had the second shot, about 20 to 30% of you probably experience some fever, chill, aches. That's your immune system ramping up. That means it's working. Many people experience that after the influenza vaccine as well. And they think, oh, the flu shot gave me the flu. And no, what it did was cause your immune system to act as if you were infected with the flu, okay? But not with the same level of danger. So our immune system responds in really complex ways. So this is an oversimplification, but it's fine. It'll serve our purposes. We have 3 different types of cells that can specifically learn what the virus looks like and remember for later. Okay, so we have B cells that produce antibodies. Antibodies can attach to the outside of the virus, they can attach to infected cells, and they call in others to kill off the virus. Okay, antibodies are really powerful. Those are B cells. Then we have 2 types of cells that are called T cells, helpers and cytotoxic. Helper T cells are what they sound like. I call them the bugle boys of the immune system. They recruit cells to come help, which is why I put arrows going out to these other types of immune cells. Cytotoxic T cells are also what they sound like, cytotoxic cell killing. They find cells that are infected with the virus. This picture is of them killing a cancer cell, but same idea. They find cells infected with the virus and they kill them off. It's fine. You have plenty of cells in your respiratory tract. You're making new ones all the time. You can stand to lose a few, and it's not going to cause you too many problems. Okay? But after it's cleared the infection with antibodies and helper and cytotoxic T cells, as well as some others that are less specific, some of them hang around. Okay? They've learned what this virus looks like, acts like. They can create antibodies that are specifically designed just for that virus, and they remember. Okay? So if you're infected again, then your immune system can ramp up and fight it off much, much more quickly than it did the first time. Okay? So when we make a vaccine, we're just taking advantage of this natural process. We're mimicking natural infection with the virus. Okay? So you get the vaccine, your immune system thinks you're infected with the virus because it looks like the virus proteins are in your body. It responds by creating antibodies, helper T cells, cytotoxic T cells can get called in, and it clears the infection that never really was. But now you have those memory cells, right? Those B cells and different types of T cells that can remember, and if you're ever infected again, ramp up and respond much, much more quickly. So the Mrna vaccines, which some of you have received, are mRNA vaccines, and they work in a really specific way, this really cool technology that's been in development for a long time. This is the first time we've really seen it put into practice. They're very quick to design, which has been great, and it's also good news when it comes to COVID variants and evolution of SARS-CoV-2. But it takes genetic material, this little mRNA strands from the virus, code for certain proteins. In this case, the spike protein, that all-important spike protein on the outside. Injects it, your cells take it up, okay? And then it makes multiple copies of that spike protein, not the whole virus, just that one protein. So there's no way you can get COVID from the vaccine, period. It doesn't, there's just no possible way. Because all you're getting is mRNA that codes for that one thing, okay? And then it promotes this immune response. You create antibodies, T cells, different types, and it can remember what that spike protein looks like. And if it sees it again, go after it. Okay. All right, but then what happens if there are variants? So the good news, even with that Brazil variant that I talked about, where it's more contagious, it seems to be reinfecting people that already have COVID. The Pfizer and Mrna vaccines generate really high levels of antibodies, more so in most cases than if you were naturally infected. And so if you're vaccinated, you might still have more protection against the Brazil variant than if you were infected for real in the months prior. Okay. So that's some vaccine good news. I like to close things up on good news. There are booster shots being generated to fight the new strains. Pfizer and Moderna, Johnson & Johnson, AstraZeneca, they've all been working on this problem for a long time. They've been following the new variants very closely and trying to develop booster shots that are maybe specific to the new variants, but some that should be able to promote antibodies against a whole host of variants. Okay, because we have some regions of that spike protein, like I said, that are conserved because it just works. There's a term for a certain type of vaccine development called reverse vaccinology, where we can look at genetic material from many different variants and say, okay, what doesn't change? What doesn't change? Because if the virus changes it, it causes some problems. What's conserved? Okay, and then we can say, well, maybe that's a great target for creating something like an mRNA vaccine for that site on the protein that doesn't change. It's like looking as if everybody in this room, we're all wearing different clothes, but we're all wearing the same socks. And we developed a vaccine that targets those socks, right? Okay. There's a fun TED Talk on that, whether you said an analogy, I really like it. All right, so there is good news. There's concern, but then it's something that the public health and the genetics community and the pharmaceutical community are all trying to work together to solve. Okay. In some really cool and unique new ways. Okay. So the last thing I want to mention here, again because it's a mixed audience, is that in all of this, over the last year and a half, the scientific process is working the way it's supposed to. But it's just different how we're absorbing it and communicating it. Okay, so normally there's scientists that come up with some kind of hypothesis, they run an experiment or an analysis, and then they'll take those results, the results of their experiment or analysis, and share them at conferences and presentations, publish them, and it's typical, every now and then one will look at the news, but most of the time it's only read and processed by people who are studying the same thing. Okay, most of us, we put out papers and 10 people we already will read it, kind of deal. But then they're taken, they're reviewed by members of the scientific community who are working on similar problems, and they ask questions, and they make comments and say, well, have you tried this? Or did you think about this angle? Or, hey, I think you might have messed up here. Did you have some mistakes? So that scientist might go back and rework it. Other groups of scientists work to collaborate and confirm the results. Okay? And then after a period of time, we'll see that science being shown as breakthroughs. You'll see more news articles about it. You'll see it start to become the basis of maybe government policy or policy within different institutions. What's different now is that the media and the government are following every single step of this process in real time. Okay, so a lot of in the past, what we've mostly experienced as scientists is there's a step-by-step progression, and then after there's some breakthroughs, that's when the public gets to know about it, government officials get to know about it, and that kind of more messy middle where things are being confirmed, replicated, people asking questions, what we think we know changes, right? Now the public is paying attention to all of that. And so it can seem like we're, and we kind of are, all in real time together, tumbling towards consensus and understanding in the face of change, right? Because we have all this evolution happening, we have vaccines coming out, we're learning and changing. So the scientific process is still very much working, but it's just kind of cracked open. In some ways that I think are good. In some ways that can impact the way the public perceives what's going on. Okay. All right. Okay, I think that's it. I'm happy to take questions. But thank you for your attention, and thank you so much for inviting me. It's been really fun.
Any questions for Dr. Sloan?
Yes.
I've got a question. I've got several that I wrote down.
Oh my goodness.
But I'll ask the one that that may apply to your public health background.
Okay.
So we're seeing all these variants that are coming out. Do you think that we'll ever truly get rid of COVID, or do you think this is going to be an annual concern, kind of similar we see with the flu? Every year they have to look at the H and the N proteins and develop a new vaccine based upon that variant, what they predict. Do you think we'll see something similar with coronavirus?
Yeah, so I think yes and no, and here's why. I don't think it's going to be quite like flu, but I don't think we're likely to get totally rid of it in the near term. We'll probably still see some small outbreaks occurring, some new variants may pop up. But with flu, because flu is continually spilling over from the aquatic bird population and from pigs into humans every year. So we can end up with these really radical changes sometimes where we have every 20 years something pandemic flu. We don't have as much human interaction with this bat population. It's more rare, there's less spillover. So that phylogenetic tree I showed you for COVID, if I showed it for flu, it would be much, much larger, right? So I don't think, and this is just Chantel Chapter 2, right? We're all trying to predict the future, and it turns out that's difficult to be a prognosticator. But I think we're going to have it be a concern. I think we'll see some small outbreaks, but I don't think we're going to need to go every year and get a new shot. Now, granted, we don't know how long the immunity lasts from the vaccine yet, so, but it might be every, 3 years you go get a booster, is what I'm prognosticating, and you guys we can put odds on the board, and you guys can place bets if I'm right or wrong, but, yeah.
I have a question. Could you speak of why the vaccination provides better immunity than actually getting COVID?
Yeah, so for a lot of people, especially since most people get mild COVID.
Repeat the question.
Oh, she wants to know why the vaccine can confer stronger immunity than being naturally infected in some people. So, especially because most people when they're infected have mild symptoms or they're asymptomatic, they may not produce as high a level of antibodies. So you'll see in the news it'll talk about antibody titers over time, and it usually takes about 3 weeks after your second shot till you've reached, your highest level of antibodies. So the amount of antibodies per whatever it is, fluid ounce of blood or something that you have. And if you only had a mild case or an asymptomatic case, then you probably still have antibodies that play at a lower level. And that immunity isn't going to last as long because your immune response initially wasn't as big, right? And so, but with the vaccine, you're getting both shots in a very controlled dose way. And it's a fairly high dose of that spike protein. So more than you might be exposed to if you only had a very mild case of COVID the first time around. And so those antibody titers per fluid ounce of blood or whatever it is, I should know, but I don't, could be much higher. The concentration is much higher than you would otherwise have. So immunity might last longer, and it might be more robust against some of these different variants. Fingers crossed. More prognosticated. But that's what it seems to be so far. Yes?
So my question was in regards to spillover, and you presented the 3 different methods. My question is, how do scientists figure out which method most likely occurred? What sort of
It's a
model?
great question. So how do we identify where the spillover occurred and when? So the answer is we very often struggle to find the moment or moments, because sometimes it's spilled over multiple times. But it's a combination of looking at the genetics of the virus and where we start to see kind of a common ancestor, that common ancestor variant. And it also has to do with where clusters of cases first emerge. So you've probably also seen in the news that the World Health Organization team just got back from Wuhan and put out a report about these different hypotheses. And they did a mix, both of those methods. They said, okay, where were all those initial cases? Can we track them back to earlier cases? Or possible cases, because sometimes it's hard to know if you don't have a sample from a patient from years ago. In relation to then where we saw that first major epidemic center in Wuhan. And then, so by combining that genetic and geographic information, you can get a sense for how things played out historically. It's not perfect. It's hard to know if it was like, Uncle Tom who ate that one dinner 3 Saturdays ago. But you can get a sense for, okay, it was probably somebody who ate some wild game from this forest 2 years ago or whatever might have been the original. Does that make sense?
Yeah.
Okay. Yeah. But they have ruled out that it walked out of the lab in Wuhan. That was actually ruled out a year ago too, because the genetic sequence of what they had there was just nothing. It was very different from what was actually spreading. And they double ruled it out because it also doesn't make sense that the case contacting, the contact tracing data doesn't match up with that hypothesis either. So it's always possible that something was missed, but that hypothesis has been pretty much thrown out as the least likely by far. It's much more likely that it's the same spillover pattern we see for zoonotic diseases, where people were interacting with wild animals in some sense. Okay? Yes?
There's been some talk about blood typing being related to certain
Uh-huh.
diseases and stuff. That's something I don't quite, maybe you can help me understand how that is. I don't really see it, can't really find the connections.
Yeah, I
Blood types.
read just a little bit about that, and I confess it's been a little while. It was something that kind of hit the news and then died down a little bit, that there's certain certain blood types that maybe confer more immunity. I think it just has to do with antigens or these markers that are present on our cells in the virus spike protein and how they bind. But I also wasn't totally swayed by the studies that suggested the blood type connection.
Yeah, 'cause I really couldn't see it. That's kind of what I teach in some of my classes about. I really can't see the
Like why?
direct connection of why O would be different than AB
Yeah.
that way.
It could also do that you're triggering my memory of something that it could also do with clotting factors, because blood clotting, because COVID has been known to cause blood clots, and a lot of people have died more because of cardiovascular issues than respiratory issues. So,
Because there's a new article that came out today about thrombocytopenia, body platelets and stuff, and maybe that's part of it.
maybe, yeah. So he's saying there's an article that came out about thrombocytopenia, which is a blood disorder, and that might be related. Yeah, so some of these blood conditions, because clotting is such an issue, could be a driving, especially early on before we understood the clotting thing. Of course, now people with more serious COVID are getting anti-clotting factors, so, which has helped a lot. But I think you and I know the same amount about that, so if you know more, find out more, let me know. Anything else? Okay, thank you.